Good morning, everyone. Welcome to the inaugural Grand Rounds of the season. My name is Alex Harris. I'm one of the Grand Rounds co-directors, along with Drs. Aditya Lati and Rebecca Mule. Next week during this time, there will be a town hall led by the chair and executive director of the Department of Psychiatry and NISP, Dr. Joshua Gordon. Before we introduce today's speaker, a reminder that if you're participating via Zoom, please post questions using the Q&A feature, not the chat feature. As the online moderator, Dr. Talati will forward questions to me for our speaker to answer. We encourage trainees to ask a question, and we'll hold space for trainee questions in person and online. If you're a trainee online, indicate so in the Q&A. Remember to fill out your CME surveys after the talk if you'd like CME credit for today's lecture. There's a QR code on the table outside for those in person, and the link will be posted on the chat on Zoom at 12:15. It's now my great privilege to introduce today's speaker, Dr. Ofer Yizhar. Dr. Yizhar is an associate professor at the Weizmann Institute of Science in Israel. He received his PhD from the Tel Aviv University, working on the molecular mechanism of synaptic function, and did his postdoctoral fellowship with Carl Deiseroth at Stanford University, where he developed some of the foundational optogenetic tools used to dissect neural circuits. He established his own group at the Weizmann in 2011. His lab develops neurotechnology for studying the brain using a combination of optical, electrophysiological, and behavioral techniques to understand the brain circuits involved in memory, decision-making, and social behavior. Recently, his lab has been focused on tools for silencing neurons and specifically for optogenetic silencing of synaptic transmission. Tools created by his lab have been broadly adopted by labs worldwide and used in a range of animal models and experimental paradigms. His work has been funded by grants from the European Research Council, the German Research Foundation, the NIH, the Israel Science Foundation, and he's received numerous awards for his work, including the Bernard Katz Prize, the Adelis Brain Research Prize, and the Suretsky Prize for Neuroscience Research. He was elected a full member of EMBO, and this past year spent a sabbatical here at Columbia as a Schaefer Research Scholar. Ofer is as thoughtful of a person and scientist as they come. The title of his talk today is New Tools to Control Neural Circuits. Please join me in giving him a warm welcome.
Thanks, Alex, for this kind introduction. It's great to be here again, actually. So I was here as a research scholar and then went back to Israel. So it's nice to visit again now and meet some old friends, meet some new people, and all the exciting science over the last couple of days. And thanks for coming, first of all. And before we start, I think we have a few kind of formalities to get over. So the first thing that I wanted to say as a disclaimer, trying to see if I can point on the right screen. Is it here? No. Never mind. So I'll use this. So I'm a co-founder and scientific advisor of a company called Modulite Bio. It's an Israeli startup that's been licensing some technologies that my lab has developed. I'll talk about the technology which we developed completely for basic science purposes. And it just so happened that people found that might be translationally relevant. So this company is doing some pretty exciting work that I'm hoping to tell you about as part of the talk. And the next thing that I was asked to state are the learning objectives for those of you who are receiving CME credit. So I was hoping that we can get through a little bit of the kind of behind the scenes of optogenetics, the mechanism of how it actually works, what it does to neurons and to synaptic connections when we apply it to these locations in the cell. We're going to talk a little bit about potential clinical applications of optogenetics, and this has been discussed continuously since the beginnings of optogenetics. There have been attempts at translating optogenetic technology, and some successes, many failures, and also the challenges in translating optogenetic tools to humans. So we'll talk a little bit about what is it actually that stands between us and being able to dream about translation applications in a variety of disorders, from neurology to potentially even psychiatry. So these are the objectives. And yeah, I don't know how you typically do this, Alex. Do you have questions during the talk or only in the Q&A at the end? Okay, great. So I started the lab in 2011, hoping to be able to put a little dent in our understanding of PFC function. And one of the, I'm sure that everybody here is familiar with the prefrontal cortex. It's a brain region that does so many things that makes it complex and fascinating to study. What you see here is a kind of overall summary of the different connections that prefrontal cortex neurons make with other parts of the brain. And you can see here that it interacts with almost every brain system that you can think of. So other frontal cortex areas, hippocampus, amygdala, striatum, and the corticostriatothalamic loop. And virtually all of the neuromodulatory systems kind of converge into the prefrontal cortex and have different effects on the different cell types and different receptor expressing cells. And of course, there's been a lot of work done on this circuit, a lot of work in animals and even in humans. And we know that certain pathways are important for certain functions, right? Which is kind of what we always aim to discover. So just to give a few examples, we know that hyperactivity of frontostriatal connections has been implicated in OCD. They have a role in working memory. They also play a role in social behavior and are implicated in social dysfunction in a variety of psychiatric disorders. A lot of work from many labs, including some of you guys sitting here. We know that connections between the prefrontal cortex and the amygdala are crucial for processes related to fear memory and are implicated, or dysfunction of these connections is implicated in post-traumatic stress disorder. We know that the hippocampal prefrontal axis, and Josh's work has been really influential in this field, is implicated in schizophrenia and is part of the mechanism for how working memory works. So a lot of these functions have been attributed to specific pathways, and a lot of this work has actually been enabled by doing precise optogenetic manipulations on these circuits. Now, if we look at the prefrontal cortex and if we try to think about maybe how we can translate these findings to modulating these pathways in a very directed way in a future application of optogenetics, we run into all kinds of problems that I'm going to kind of bring up through the talk. But one of the more important issues is that if we look at the PFC as a whole and we look at all the neurons there, we know that, for example, different neurons can project to different places. These can be completely genetically identical neurons. And if we wanted to dream about manipulating a certain pathway to treat a particular disorder, whether it's schizophrenia, OCD, or whatever disorder we talk about that's been associated with this particular pathway, it's very difficult actually to single out a particular cell population like we can sometimes do very easily in animals, right? We can get a mouse that expresses a certain recombinase in a particular group of neurons. Sometimes even in mice, we don't have this genetic identity associated with neurons projecting to region A and not to region B. So if we wanted to manipulate the pathway, the way that it makes most sense to do it is not to manipulate the neurons themselves, but actually to control the pathway, right? The outputs to the distant region, because if we single out these axons, we can actually manipulate that particular projection specifically and not have to worry about targeting these particular cells in the circuit within the cortex, right? So that's kind of a long-term goal of everybody doing optogenetics in animal models that are not amenable to this kind of genetic manipulation. Another problem is that a lot of these cells are not going to be projecting just to one target, right? Cells have collaterals. Cortical cells have sometimes many collaterals. And it could be that if we activate these axons, we also activate their projections to the other regions. So we kind of lose the specificity by triggering action potentials in the axon. And in fact, if we look at a single cortical neuron, and this is an image from a mouse from a paper by Mike Economou a few years ago, you can see the cell body of the neuron. This is a motor cortex neuron sitting here in layer 5. And these are all the axon collaterals that this neuron has. So these neurons sometimes have multiple collaterals to multiple different circuits. So it's not enough to modulate the neuron itself. It would be much more beneficial to modulate the axons in the pathway that we need to target. So one thing we can do with optogenetics, which I'm sure you guys are all familiar with, is to activate the axons, right? We can express channelrhodopsin in the neuron. The channel rhodopsin will make its way through all of these collaterals, and we can specifically trigger spikes in these axon terminals and get synaptic release at the target. The problem with this approach is that this spike will most likely also propagate back to the soma, right? And from the soma, it can just as easily propagate to all the other projections of the axon. So in a way, activation of an axon with optogetics might be site-specific if we work in a slice preparation in which the axons are severed from the cell body, but in vivo in an entire animal, it might actually end up not being very specific. On the other hand, if we switch to silencing, and as you know, optogetics can be used for bidirectional manipulations, if we silence a particular population of terminals, we're much more likely to contain the silencing within that area and that presynaptic projection and not have it, for example, silence the axon collaterals going to another location, right? So far, I hope that this makes sense. If not, happy to discuss this. So excitation is very easy to do. Inhibition turns out to be much harder. And this is for a number of reasons. So pathway-specific silencing, as I argued in the previous slide, is crucial for understanding what these projections are doing. So both for basic science to understand the role of a projection in a particular behavior, it's much more interpretable, I think, to silence a particular population of axons. Inhibition is also more translationally relevant, and that we can discuss at the end of the talk, because a lot of neural disorders are actually associated with hyperactivity of a particular cell population or projection. But the trouble is that inhibition requires much more than excitation. If you think about it, if I want to trigger spikes, let's say at 20 hertz, our favorite frequency, in a particular axon population, all I need to do is to deliver these really short pulses of light, and I can trigger these spikes very efficiently with a minimal amount of light. If I want to silence ongoing spikes, I need to kind of blanket inhibit the projection for the entire time because I don't know when the spikes are going to happen, right? So the light requirements for inhibition are much more stringent than for excitation. And this comes with another problem, is that the inhibitory optogenetic tools, at least the older kind of microbial opsin-based tools, are much less light sensitive than the channels. And this comes from the fact that these tools are essentially pumps, and pumps will only pump one ion per one photon that actually activates them. So the stoichiometry of ions per photons is 1-to-1, whereas for a channel or for G-protein coupled receptor, the stoichiometry is much more in favor of these newer opsins. So this kind of problem we were facing in the beginning of our work in the lab. And when I started the lab, I told you I was interested in modulating the prefrontal cortex and specifically the outputs of the prefrontal cortex to different circuits. And we were thinking, okay, we have these inhibitory tools. We can do this in the soma. Let's just do the same thing in the axon. And then we solve the brain. We solve what the prefrontal cortex projections are doing. And this led to an entire kind of tangent in the lab that we call optogenetic adventures in the presynaptic terminal. We started out by just testing what happens when we deliver light instead of to the soma to silence the neuron, to the axon terminal to silence that particular pathway. And here we discovered some really shocking things. So for example, we discovered that halorhodopsin, which is the kind of canonical inhibitory optogenetic tool that maybe many people have been using, very efficiently pumps chloride into the neuron. This is the mechanism of action. It's a pump, it pumps chloride. It does the same thing whether it's in the soma or in the axon, but actually in the axon, the degree of silencing that we could evoke with this chloride pump is not that great. We could reduce the evoked EPSC amplitudes by about 20%. And we wanted more because we wanted to at least partially or at least halfway silence the release so that we have robust behavioral effects. So we switched to another inhibitory optogenetic tool called ARCH. This is a proton pump that pumps protons from inside the cell to the outside. So essentially it's hyperpolarizing just in the same way that pumping chloride is into the cell. And we found that ARCH actually more efficiently silenced evoked release when we record from postsynaptic cells. You can see that over time, it kind of accumulates activity and it brings us down to about 60% reduction in transmission, which seemed to be pretty great. On the other hand, if you look at spontaneous transmission from the same terminals, and this is something that Josh and I discussed a lot, we found that during the same time that evoked transmission is silenced, we have an increase in spontaneous release from these terminals. And this was really paradoxical, very frustrating. We struggled to understand why this was happening. And eventually what we found is that this proton pumping from the intracellular to the extracellular side actually changes the pH inside the terminal, which isn't kind of very surprising, but you don't think about it when you're just applying optogenetics. It's not really changing anything in the soma, but in the terminal where you have pH-sensitive calcium channels, what you get is influx of calcium when the pH changes inside the terminal. So this is what's causing this gradual increase of spontaneous transmission. And because we wanted to find something more efficient, we want to look at chloride channels. So channels, as I told you, are better than pumps because the stoichiometry is much more favorable. We get one photon and many ions come through the channel. But it turns out that if you express one of these light-activated chloride channels, so these are called ACRs, or anion conducting channel rhodopsins, you can get very nice silencing of the soma, but if you shine light on the axon, you actually get a spike. And this is because chloride in the axon is much higher than in the soma. So what you have instead of chloride flowing in is actually chloride flowing out and a depolarization. So it seemed like whatever we tried, we could never get efficient inhibition. We always got efficient excitation of the axons. It was very frustrating. So Matthias Mann, who was a student in the lab at the time, decided to try and fix this problem or try to find a better tool to do this. He was actually the one who discovered this paradoxical excitation with all these other tools. And we said, okay, let's try to solve this. And we reasoned that if we target G protein coupled receptors at the presynaptic terminal, we might be able to get a clean inhibition, essentially like the brain does it naturally, right? We have a lot of different GPCRs expressed in the presynaptic terminal membrane. And when these receptors are activated, for example, as you see here, this GABA B receptor expressing on the terminal, when it's activated by GABA, the G protein dissociates. It activates or it silences the activity of the adenylyl cyclase. It reduces cyclic AMP. And at the same time, the G beta gamma subunit does two different things which dramatically reduce synaptic transmission. It closes voltage-gated calcium channels and it directly clamps the snare complex, which mediates vesicle fusion. So by activating this single receptor, you get multiple activities that are all kind of contributing to a reduction of synaptic transmission. So the idea was, let's try to find a light-activated protein, an opto-GPCR, that'll couple to this endogenous pathway. And there we have several benefits, right? We're both activating an endogenous system that's naturally inhibitory, and we don't have this paradoxical excitation, presumably. And at the same time, we also get amplification, which G protein coupled receptors do. So one activation of the receptor can trigger multiple cycles of silencing. So potentially we would have a much more sensitive or light-sensitive actuator. We looked at the range of existing rhodopsins in nature, and there are a lot of rhodopsins that we use, many animals use for vision. These are called visual rhodopsins. They're all GPCRs, but most of them, or all of them, are bleaching rhodopsins, right? Once we activate them, they lose the retinal chromophore, and you need to reconstitute the rhodopsin by binding another retinal chromophore. This works, very efficiently in the retina, but it doesn't really work if you're activating the rhodopsin deep in the brain in some neuron population. But there actually is a group of type 2 rhodopsins that are also GPCRs, also light activated, that are called bistable rhodopsins. And these rhodopsins operate in a different manner, so they never release the retinal chromophore. It just switches conformations and then switches back, whether spontaneously or with a different wavelength of light. And there are a number of examples from these really unique rhodopsin groups. One's called OPN3, and this can be found in many organisms, including even us humans. There's an OPN3 expressed in various tissues in our body. And that's actually still a mystery what exactly it does, because a lot of these tissues are never exposed to light, like the liver or the kidneys. But parapinopsin, for example, is an opsin that expresses in the parapineal region. In fish, for example, it's related to cycling, light-dark cycling of the brain. And some of these are actually bistable. So we started trying to express these opsins and figure out what they do when you take them and express them in mammalian neurons. There was a single paper from a Japanese group that showed that a couple of these opsins, the pufferfish TMT, multi-tissue opsin, and the mosquito OPN3, which is from this OPN3 group, are actually able to reduce cyclic AMP when they're activated. This is cyclic AMP measurement in HEK cells. So we started with these, and we were just gonna test them in neurons and see whether they're capable of silencing transmission. And one of the benefits of OPN3 is that it's actually a red-shifted opsin, and we'll get to that a little bit later, but red light is always more beneficial to use than green or blue light because it's less damaging to the tissue, lower energy photons. We found that if we express the mosquito OPN3 in mammalian neurons, we can actually evoke GERK-mediated currents. These are potassium channels that are activated by GI GPCRs. And this was already a really promising indication that this might actually work in neurons. We also find that if we record synaptic transmission in cultured neurons, we can cause a really dramatic suppression of synaptic transmission just by shining a single pulse of green light on the neuron. And this indicates that this opsin has long-lasting properties, which are actually beneficial for our applications with optogenetics. We can use much less light, we can pulse intermittently, and get this consistent inhibitory effect. So as a result of this long-lasting activity, OPN3 is actually very light sensitive. This is a light sensitivity curve plotting the light response or the effect on synaptic transmission versus the irradiance of light that we use. So just by comparison, these are a few of the kind of canonical optogenetic tools. These are halorhodopsin, archirhodopsin. This is a channel that we published a few years ago. It's a chloride channelrhodopsin. And this is OPN3, and you can see that the shift to the left is about 4 orders of magnitude. So this means that this opsin is about 10,000 times more light sensitive than our existing inhibitory opsins. And this results partly from the fact that it's a G protein coupled receptor, it amplifies the signal, but also from the fact that its activity is more long-lasting than the microbial opsins. We tested it in a bunch of different preparations. This example is from the mouse visual cortex, where we expressed OPN3 in the visual thalamus, and we recorded in visual cortex in a mouse that was awake and looking at a screen where we could display visual stimuli. These are responses of neurons in visual cortex to this visual stimulation. You can see here all of these neurons are significantly modulated by the visual input. And this is after illuminating the cortex with green light, and you can see that most of these responses are gone. This is because we're suppressing the activity of these thalamic terminals, delivering the visual information to the cortex. This is another way of looking at it as a PSTH before versus after illumination. And the effect is relatively long-lasting, so this single pulse of light caused a suppression of the evoked activity, which recovers with a time constant of 5 minutes. So this is relatively long compared to conventional optogenetic tools, but it's also much shorter than other, if you think about pharmacological manipulations or chemogenetic manipulations, which can last for hours. This is still relatively time-resolved. Now, we know that red light is beneficial because we all did the experiment where we took a laser pointer and put it on our thumb, or in this case, on our cat. And red light just propagates really well through biological tissue. And because OPN3 was red light sensitive, we wanted to check whether it can actually be used with red light in a way that maybe even permits non-invasive optogenetics, which would be huge, right? We talked about potential clinical applications. This would actually be really useful. So Jonas, who was a postdoc in the lab, decided to test that. So he used this basically edge of the action spectrum of OPN3 and asked, what's the minimal amount of light power that we can use in the red to activate this opsin? And I won't spend too much time on these recordings, but basically he found that even at a very low light intensity of 20 microwatts per millimeter square, which is a very low irradiance, you guys using optogenetics probably use 10 milliwatts or so for any kind of silencing or even stimulation. So this is a really minimal amount of light. You could get very efficient suppression of synaptic transmission. And this is the curve basically plotting the progression of silencing with a continuous light at these different irradiances. So you can see 200 microwatts almost instantaneous versus 20 or 3 microwatts even. You can see the accumulation of the effect over time. So you can basically deliver a very weak amount of light but extend the light pulse, and you get an accumulation of the inhibitory effect over time. Daniel was working on another project related to oxytocin in pups, which I talked about last year in the neuroscience seminar that I gave a talk in, so I won't go too much into it, but basically just to tell you that he was able to actually get red light, to activate OPN3 all the way down in the hypothalamus of these animals, simply by placing a cover slip on the skull without even removing the skull, but just replacing the piece of black skin, which obviously absorbs a lot of red light. And you could see reduction in CFOS expression in the oxytocin neurons in which the opsin was expressed in the hypothalamus, simply by putting a projector with red LEDs above the cage in which the mouse was sitting. So this allowed a completely new range of experiments in the lab. We're interested in early life social behavior, and this really allowed us to do optogenetic manipulations in pups without implanting any hardware and without tethering them to the optical hardware. And he was able to show, for example, that activating OPN3, so silencing the hypothalamic oxytocin cells, changes the dynamics of vocalizations, whereas a mouse that's isolated from its dam and litter will cease vocalizing over time during isolation. These mice in which we silenced oxytocin didn't seem to be calming down in the same way. So we have this whole story that's actually coming out next week. So if you're interested, you can look for it. Another thing that we can do now that we have a really sensitive inhibitory tool is to have an implantable recording device that includes both the neurologger, so allowing us to record through these electrodes, but also a tiny LED which delivers light through this optical fiber. And because of battery constraints and spatial constraints, we couldn't put a really strong LED in there. We could only deliver 90 microwatts per millimeter square of light. But as you saw before, 90 microwatts is actually enough to get robust suppression of activity in these cells, completely wirelessly recorded and modulated. So yeah, this is just one of these social behavior experiments where we can silence PFC using these wireless neurologgers. So why is light sensitivity important, not only for basic science, but also potentially for translational purposes? And this is because if you think about it, in the mouse brain, we can put an optical fiber, and many of us do this regularly these days. We can express our optogenetic actuator, and any cell which is within the range, this blue circle basically defines, I think, 0.1 milliwatt per millimeter square. All of these neurons that get enough light will be activated or suppressed based on which opsin we express. And this is really easy to do in the mouse brain. Here, we're basically capturing almost an entire hemisphere of the cortex by just putting a single fiber. If you try to do this in a larger brain, the fiber stays the same, the light spread stays the same, but in this case, this is a monkey brain. This is a figure from Ilka Diester's paper in the macaques, first paper showing optogenetics in macaques. And what you can see is that the same light propagation diagram will only capture not even the entire depth of the cortex. So doing this kind of translating this kind of optogenetic manipulation from mice even to monkeys is going to be really difficult because we're catching a much smaller fraction of the functional region that we're interested in modulating if we have a low light sensitivity tool. If you take it even further to the human brain, then this is what it looks like. So over there, you can see the little circle capturing the entire hemisphere of the mouse brain. This is what it would look like on a human brain. So here we're capturing a tiny, tiny fraction of a functional region of cortex, right? So if we wanted to do anything that's translational, directly translating optogenetics to the human brain, this would be a major caveat. And indeed, if you talk to some of the people who were involved in the early startup companies that were interested in translating optogenetics to the clinic, this is the first thing that they were running into. They were having really nice behavioral effects in animal clinical models, but these behavioral effects would not even be possible in a larger brain because the tissue would be, orders of magnitude larger. And even in these animal models, they started seeing photo damage after a while, more than an hour, for example, of delivering continuous light to the tissue. So OPN3, as I told you, is extremely light sensitive, right? It's a 10,000-fold increase in light sensitivity compared to the conventional optogenetic tools. And this is why it was interesting for people who were thinking about potentially translating optogenetic therapies. So Modulite, the company that I'm involved with, and already told you that I have an interest, so you can take this with a grain of salt. But I was interested in showing this because they've been taking this tool, this opsin, making AAVs that might even potentially be usable in humans, and looking at whether they can get therapeutic modulation of certain phenotypes related to neurological disorders. So the first experiment that we did together was looking at whether we can suppress epileptic activity in the brain of a rodent for this for now. What they did was to inject kainate into the hippocampus unilaterally, but before that, they injected the opsin expressing AAV, and this is what it looks like. So you can see this rat brain. This is unilateral expression of OPN3M scarlet in the hippocampus. Into the same place, they injected kainate to generate this chronic model of epilepsy. And they decided to do transcranial illumination. So what they did was just to put a little chip that includes red LEDs. I think at this point it was 8 red LEDs just on the skull. They measured the light. They saw that at the hippocampus, they have plenty of light to activate OPN3. So the expectation was that if the hippocampus becomes epileptic, activating this transcranial illumination would actually be able to suppress the ongoing seizure. And this is one of the many examples where you can see a rat undergoing a seizure and then light switches on. You can see that it takes a while. And as you saw before, the effect of the opsin takes a while to activate. But then after that, the seizure terminates and the rat continues behaving normally. This is, of course, kind of cherry-picked video, but you can see the overall results. So if we look at seizure score, you can see that OPN3 during light reduces the seizure score. It also reduces dramatically the event duration of these seizures. So when they're targeting the seizures when they're happening. This is over time, so multiple periods of light and dark. You can see that these animals are showing modulation of the seizure score, where the control animals that are injected with GFP are not. And this is, again, seizure duration. You can see that there's a dramatic modulation here compared to the control animals. So this indicates that this inhibitory opsin, that's a GPCR, can actually be used to suppress or to at least reduce seizure activity in the hippocampus. This is in rats. The next, this is just a quantification of the light versus dark ratio. You can just see this again. Same data plotted differently. So the next question was, okay, this was in rats. We know that optogenetics works great in rodents. Can it actually work in a larger brain? And it's very hard, of course, to do a seizure model in primates, but we started collaborating with the lab of Greg Worrell at Mayo Clinic in Minnesota. And they've been developing this pig model of epilepsy where they do pretty much the same thing that we did in the rats, but they do this in Göttingen pigs, micropigs. And the idea was to do exactly the same experiment, basically inject the opsin, get sufficient expression of the opsin in the entire unilateral hippocampus and repeat the same experiment and see whether this would really work in an animal in which the hippocampus is closer in size to a human hippocampus. So this is what it looks like. They do everything under MR guidance and also the virus injection. You can see the virus is co-injected with gadolinium. So you can actually visualize that it's in the right place. You can then do histology afterwards and see the opsin expressing in the hippocampus. And then the next thing they did was to implant recording electrodes next to an LED array or an optical fiber. We had 2 different variations of the experiment. And then the next question was, can this activation of this inhibitory opsin suppress evoked activity in the hippocampus? This was without inducing epilepsy. So what you see here are basically experiments where we stimulate in the anterior thalamus and record in hippocampus. And you can see this is the evoked activity in the control condition. And then we illuminate with green light and we record again. And you can see that the evoked activity diminishes. This is the summary in one animal and another animal in which we repeated the experiment again after a few months. And then the next question was, does this actually allow suppression of seizure activity? And the answer is essentially yes, but so you can see here that kainate injection triggers a seizure, which is shown here as the about 30 hertz activity in the spectrogram. And then illumination suppresses this ongoing activity. Afterwards, when illumination is terminated, you can see the seizure basically picking up a bit up again and also generalizing to other parts of the brain. The yes but was because as kainate diffuses out, it becomes more and more difficult to actually control the seizure. So this was a nice proof of concept demonstration, but there's still work to do to figure out whether we can really control the generalization of the seizure after kainate diffuses out. Another proof of concept experiment, which was, to me, I think, pretty amazing, is looking at essential tremor. And essential tremor, as you probably know, is a pretty devastating neurological disorder. It does have all kinds of treatments, both pharmacological and surgical. So people do removal of parts of thalamus, for example, or deep brain stimulation. But we wanted to see whether we can gain from the knowledge that's been accumulated about the circuit basis of this disorder to really see if we can modulate it based on this knowledge. So these are drawings of a patient before and after surgery of essential tremor. You can see the tremor activity in this spiral that they're trying to plot. What we know about the tremor in this particular disorder is that it originates from the cerebellum and propagates to the thalamus and the cortex, and then to motor system, basically. So the earliest point at which this oscillation is observed is in the connection between the Purkinje cells and the deep cerebellar nuclei. So there's an oscillation that's formulated in this loop in the cerebellum, and then it propagates out to the thalamus and the cortex. So the idea was, let's see if we can express the opsin in the Purkinje cells, target this projection by putting light into the deep cerebellar nuclei, potentially suppressing the activity of these synapses enough so that it stops this loop from generating this tremor activity. And what you see here is a mouse that was injected in this way. So the fibers are going to the deep cerebellar nuclei. You can see that it's expressing tremor because it was injected with harmaline. And as we illuminate this projection, basically we can completely eliminate the tremor activity. So this is just one example. What you see here on the back of the mouse is just an accelerometer to measure the tremor activity. We can generate these spectrograms. You can see here harmaline injection, tremor starts and kind of stabilizes at about 10 hertz oscillation. And then as we deliver light into this circuit, we get almost complete elimination of the tremor activity, and it recovers after a couple of minutes of no light. You can compare this reduction of peak amplitude in the spectrogram. So this is before, during light, and after light, where you can see partial recovery, to a Prax 944, which is a drug that's now undergoing phase 3 clinical trials for tremor. And you can see that the suppression by the drug is significant, but not as robust as the suppression that we have here. So this indicates that by targeting the correct circuit, we might be able to use optogenetics in a way that is more precise and potentially also more free of side effects than any of these pharmacological agents. Of course, there are problems, right? But I'm painting the pink picture now. So what I've been referring to basically is direct translation application of optogenetics. And this is from a review paper that I've been working on with Christian Luscher, that I'm sure that some of you are familiar with, and Carl Deiserath, where we try to think about what are the challenges in translating optogenetics to the clinic in this manner. So I'm sure that a lot of insight from optogenetic experiments will come to therapy from just a better understanding of the circuits. And honestly, I think this is what we still need, like especially in psychiatry. I think we need to better understand the circuit dysfunction that is triggering the phenotypes associated with these diseases, right? Before we can do any kind of, clever manipulations. But if we do have a circuit candidate, we can actually think about how to directly translate these insights from the rodent brain and potentially other animal models into human translation. And is this even remotely possible? So the simple answer is yes, it's actually already being done in humans because there are clinical trials now where optogenetic tools are being used, injected using adeno-associated viral vectors into humans. And this is the first paper that demonstrated that it's actually feasible. So this is a paper from 2021 by José Sahel, Botan Rosca, and others, where they showed that if you inject chanorodopsin into the retina to express in retinal ganglion cells, you can actually trigger the activity of the retinal ganglion cells, even if you don't have any photoreceptors in the retina. So this is retinal degeneration. It's a pretty common disorder. And they showed that if they put this opsin into the retina of the human and they couple it with a goggle, which helps to compensate for the fact that the opsins are not as light sensitive as the endogenous system in the retina that detects light. So they have to actually use the camera and a projector to project light into the retina and basically generate the image from scratch, basically. Then these patients report being able to see objects, being able to walk outside in a light. And apparently it's even more effective as the younger the patients are, because the brain undergoes some kind of learning process to figure out how to deal with these signals that are most likely not remotely similar to what the brain gets from the retina, which is behaving naturally. So if we think about the gene therapy aspect of optogenetic therapy, then it actually already has been demonstrated to be not so much of an obstacle. And these patients, I think the earliest patients that to be injected with AAVs in this way have already been expressing for more than 6 years, and they still haven't reported any adverse effects simply of the expression of chanorodopsin in these neurons. It's never been done in the brain before or in the peripheral nervous system, so this is still a challenge. So how would it work? And this is really kind of to stimulate your imagination and generate a discussion that I'm sure would be interesting. The idea would be that we either inject an AAV directly into the brain like we do in our animal models very routinely. This would cause very focused expression of the opsin in a particular circuit. Or potentially we could use these new variants of AAV that have a tropism to the brain, so you can inject them systemically and they somehow manage to get across the blood-brain barrier, and they have a tropism to neurons, so they will mostly express in the brain. This has all kinds of problems, side effects, liver expression can trigger all kinds of issues. So I think the direct injection into the parenchyma might be more feasible, at least at this point in time. You could also think about potentially using optogenetics to modulate pain by expressing in peripheral ganglia. And we know that there are many pain disorders that are completely lacking a cure, cluster headaches, severe migraines, things like that, where people undergo injection of Botox or other kinds of pharmacological agents directly into the ganglia. So this might be a way of simply injecting the AAV once and then illuminating every time the pain needs to be managed. And then in terms of delivering light, there are many different ways that are potentially possible. We can use LED arrays, maybe similar to the one that I showed you that we put above the mouse. We could modify DBS probes to emit light instead of electrical current, or we could even go for the transvascular approach, just like they do stents, intravascular stents, through which you can actually record neural activity. You can think about potentially developing optical intravascular stents to deliver light. So there are many options. This is all completely hypothetical, but this is just a way of trying to imagine what might be possible if we pass all of these scientific and also regulatory hurdles that are waiting for these approaches down the line. So I'll just summarize and we can have a discussion. We found that modulation in the presynaptic terminal with optogenetics suffers from all kinds of unique constraints, and we're trying to find ways to overcome these constraints. This led to the development of these bistable GPCR rhodopsins that are actually now pretty popular. We put these opsins on Adgene, maybe some of you are using them. They seem to be really effective in animals where you want to particularly silence a particular projection. So OPN3, the mosquito-derived OPN3, which is the reason for this cartoon that some of you might have been wondering about, allows redshifted, highly light-sensitive presynaptic terminal silencing, and enabled a preclinical proof of concept set of experiments in which we managed to reduce the severe symptoms of neurological disorders in rodents. Yeah, and we discussed all kinds of issues related to direct translation of optogenetics in CNS and PNS. I'm really looking forward to hearing what you guys think about all of this. So just to take a minute to thank the people who did the work. So this is Matthias, who led both the discovery of the artifacts related to trying to silence synaptic transmission, and then also developed the mosquito OPN3 as a tool for optogenetics. Jonas, who developed another tool based on this early work and recently published. Daniel, who did the work in PUPS with the red light. And this is the Modulite crew, which we've been working with on trying to understand better what are the pathways to direct translation of optogenetics. This is our funding, and thank you all for your attention.